And today I have the pleasure of introducing Dr. Alexander Bashmanov. So Alex is a scientific leader in the Global Comparative and Translational Sciences Group in GSK, Pennsylvania. Previously, he was the senior faculty member of Monell Chemical Sciences Center in Philadelphia, USA. Alex received his degree in veterinary medicine from St. Petersburg Veterinary Institute in Russia, and his PhD in biological sciences from the Pavlov Institute of Physiology. Thereafter, he completed a postdoctoral fellowship at Cambridge University in UK. And today, we have the honor of having Alex share with us the biological mechanisms involved in sensory perception of medicinal palatability. And with that, I will pass the stage to Alex.
David, thank you very much for introduction. Hello, everyone. And just on a technical side, I want to make sure that I have control of slide advance. Mahalia, can you hear me? Okay, yes, I do. I'm very excited to present this webinar because it gives me a chance to share my past background in basic neuroscience of chemical senses. And since I joined GSK 2 years ago, I have developed some relationships with people who formulate drugs. And in our discussions, we come up to some questions where I felt that better communication or better crosstalk between practitioners and basic scientists would benefit both groups. And this is actually a motivation and for me to present today and the goal of my webinar. I will go over a few themes in my presentation. I will start with just introducing some basic concepts to point out where neuroscientists and practitioners of drug formulation may differ and how they use language and what concepts they discuss. I will update you on basic mechanisms of chemical senses which influence drug palatability. The main chemical senses are taste, smell, and chemesthesis, and discuss what do we mean when we say flavor perception. And finally, I will go over some practical questions which drug formulation practitioners encounter, how they can be guided by knowledge of neuroscience. These questions are how and when to assess medicine palatability, and if there are issues with palatability, how to mitigate it. Well, medicine palatability is a real issue to some medicines, actually to quite a lot of them, and also the focus of this working group and probably interest of audience in pediatric medications. Medicine palatability is not limited to medicines to children. It's also applicable to medicines, for example, for senior people or geriatric population and for inhalation medicines. Regulatory agencies pay attention and require some taste assessment to guide acceptability and preparation of formulations. And what I'll try to do in my seminar to bring knowledge of neuroscience, how to explain how taste works and how it could help to solve practical problems of drug formulation. And I'll start with just this question: do we, to understand each other for neuroscientists and drug formulations, we need to make sure we use the same language, and we do use the same words, and the terminology for neuroscientists shown on the right and drug formulators shown on the left does overlap, but this overlap is only partial. And what I noticed that often these two groups of people assign different meanings to the same terms, and we'll try to straighten it out during my presentation. So what do neuroscientists mean when they say chemical senses? It could be a broader or narrower kind of type of mechanisms, but the main chemosensory systems involve taste or gustatory system shown on the left, smell or olfactory system shown in the center, and kinesthesis, which is mediated by somatosensory system. And throughout my presentation, I will come back to this images, and we will get better understanding of mechanisms and stages of processing of this chemosensory information and how different channels interact. One thing which is quite clear from this system, if you overlay the three chemosensory systems, smell, taste, and kinesthesis, you will see that there is substantial or anatomical overlap both in oral and nasal cavities. And this overlap, this anatomical and functional overlap among these chemical senses is what drives this concept of flavor as complex perception. One way to illustrate flavor is kind of to decompose it on some example. Like say, imagine you put in your mouth a piece of food, like for example, strawberry. You will start detect orthonasally through your nares smell of the strawberry even before it hits your mouth. When the strawberry is getting in your mouth, you will start perceiving taste of the strawberry, which could be sour and sweet, probably. And then some odors retronasally will go through the back of the mouth and also will stimulate olfactory epithelium. So when we'll say we'll taste strawberry, it would more accurately described by neuroscientists, you would perceive flavor of strawberry, which is composed of smell of strawberry and taste of sourness and sweetness. But flavor is not limited to this factors. There are more compounds which could contribute to flavor, like irritation, touch, and temperature. And in case of strawberry, we probably would pay attention how soft or hard it is to chew, or what is surface of this strawberry, how smooth or rough it is. And it's a quote from Wikipedia, which is a good way to see what a lot of people mean by particular term, and I guess it's consistent with flavor being defined as complex sensation involving taste, smell, irritation, temperature, and texture. And particularly later in my presentation, I'll touch bases about sense of touch, no pun intended, which is quite important for palatability of dispersions of oral medications and contributes such sensations as mouthfeel, texture, grittiness, and chalkiness. One simple message to take from this slide is that flavor is not exactly the same as odor. Flavor is much more complex sensation. Now, what I noticed that flavorists, or called flavorists, people who professionally deal with flavor, and neuroscientists use flavor a little bit differently. For flavor, it's typically a descriptor of particular compound or formulation. For neuroscientists, flavor typically used when sensation can be attributed to any single of chemosensory mechanism, and it's more complex interaction among those mechanisms. And the other way to kind of see how flavor mechanisms are treated by practitioners and neuroscientists, I try to depict in this picture. Typically, flavorists would think about biological system processing flavor information as a black box, and focus would be on manipulating stimulus and see what are results in terms of perception. What neuroscientists do, they try to decompose this process in specific mechanisms. And what I will try to show in my presentation is that actual knowledge of this mechanism, neuroscience, neurophysiological mechanisms of flavor perception could help solving practical flavor problems. What is exactly perception when we say flavor perception, for example? And again, it's quote from Wikipedia, which links perception to processing of sensory information. And sensory systems are the way we create perceptions, and typically they have very uniform organization. When stimulus hits some receptors and this information is conveyed through afferent nerves to the brain. There are several main sensory systems which convey senses like vision, hearing, touch, temperature and pain, taste, smell and balance. And several of those systems are actually able to detect chemicals, and that's why they are called chemosensory systems, or systems responsible for chemosensation. And this is a familiar image. You can see that all three of these chemosensory systems, olfactory, somatosensory, and taste, have receptor cells or nerve endings at the end. They have nerves and they convey information to the brain. And when information is processed from stimulus to the brain, there are several terms which neurophysiologists use. One is transduction, and transduction typically happens at the receptor cell or at the receptor when, say, chemical stimulus is converted into a biological response. The other term is transmission, and it means that information from a receptor or receptor cell is transmitted to the afferent nerve. And all the system of converting information about stimulus, in case of chemosensory system, it's chemical stimulus, converting this information into pattern of neural responses is called coding. And when this information reaches a brain, we are able to perceive or consciously detect this information, and this leads to behavioral responses, and many of these behavioral responses are actually very important for practical aspects of medicine, such as acceptance, consumption, or rejection, which may influence medication compliance. So when we talk about chemosensory perception, we have several aspects or dimensions. One is modality. For example, taste and smell are two different chemosensory modalities. Within each modality, there are different qualities. For example, within taste, there is sweet and bitter taste qualities, and there are a large number of modalities within smell. The other dimension is hedonics, whether it's pleasant or palatable or unpleasant or aversive, and intensity, whether our perception is strong or weak. And just as term implies, medicine palatability assessment primarily asks question whether medicine is palatable or aversive. It's focused on hedonic perception of stimuli. And the important thing to remember is that it's not the only perception from medicine which we can detect. Take-home message from this slide is bitter is not always aversive. Often it is, but aversion could be caused by other factors, for example, irritation, and bitter is not always aversive. For example, we can learn liking bitterness of beer or coffee, at least some of us. Well, there are a lot of factors which can affect chemosensory perception, and I could probably talk an hour about each of them, but I wanted to just briefly mention that age is an important aspect which influence chemosensory perception, and it is particularly important for pediatric drugs. Just examples of what's the difference between children and adults. Children different how much they like sweet, sour, and salty tastes. Typically, they like them more than adults. Children, as I just mentioned, don't have learned responses to some bitter compounds, which many adults have, like, for example, beer or coffee. And these learned responses to learned liking to bitterness may influence responses to other bitter tasting products, for example, medicines. Children, when we want to test taste perception, children often may not be able to perform the same sensory assessment tasks as adults do. And when children perceive taste, they may not always be able to report how they sense it. So something to remember is children are little humans, but they are not exactly like adults in terms of their sensory perception. The next few slides just will give you some basic facts about how taste system works. And the main taste organ is tongue. The tongue has several taste papillae, circumvallate papilla in the back of the tongue, fungiform papilla in the front of the tongue, and foliate papilla on the side of the tongue. If we take a histological section and look under a microscope at this papilla, we will notice that their shape is somewhat different, but all of them have a similar structure, which are shown by these black dots, which are called taste buds. If we increase our resolution and zoom into these taste buds, we will see that taste bud is composed of tightly packed taste receptor cells, which have apical ends facing to the taste pore, and that's where taste stimuli interact with the taste cells. And the basolateral ends of these taste cells connect to the nerves, which send information to the brain. These taste buds are quite complex. They can consist of different taste bud cells with different properties. And one important type of cells is so-called type 2 cells, which include bitter responsive cells, which are main targets for drugs causing aversive taste. So how does bitter taste works? Bitter taste receptor cells are a subset of type 2 cells. They are distinct from other type 2 cells which respond to sweet or umami taste stimuli. And these bitter taste responsive cells respond to bitters because they express bitter taste receptors, and they do not express other receptors, for example, for sweet or umami taste, or for salty or sour taste. Bitter taste receptors are called TAS2Rs, which is abbreviation from taste receptors type 2. They are G protein coupled receptors. They are built in the cell membranes, have 7 transmembrane domains, extracellular and intracellular space. There are each species, including humans, typically has multiple TAS2Rs, bitter taste receptors, and single cell express many of these taste receptors. Humans have, the consensus is that humans have 25 functional TAS2Rs, bitter taste receptors. Also, total number is a little bit larger just because some of them are non-functional. Different species differ quite widely in number of taste receptors, from just a few in fish and some birds to 50 something in frogs. And this is important to remember, that if we want to use animal models or some other species to predict human responses, this difference in TAS2Rs may limit may put some limits on how predictable are data from these systems. So there are many bitter taste receptors. There are even more bitter compounds. How do they match? And this question was addressed using heterologous expression of taste receptor system using heterologous approach, when taste cell lines, typically so-called HEC cells, are transfected with plasmids encoding different TAS2Rs, and they are tested for ability to respond to compounds. It was shown that each of the 25 human TAS2Rs differ quite widely in how many different compounds they could respond. Larger circles show a larger number of compounds which activate particular receptors. And some receptors are still orphans. For them, no compounds which activate them are found. Many drugs are bitter, and they do activate TAS2R receptors. This is a kind of subset of data from a seminal paper of Wolfgang Meyerhoff and his laboratory, who characterized large number of bitter compounds with and their activation how the ability to activate taste receptors. So the compounds with pharmacological properties are shown on the left in as columns, row headings, and each column shows different TAS2R receptors. And you can see that for each TAS2R and for each bitter compound, there is unique pattern of activation. And in some cases, single drug activates just single taste receptors, the column saying NR shows number of taste receptors. In some cases, a bitter tasting drug could activate multiple taste receptors, like say for up to 15 for diphenidol. And in some cases, bitter tasting drugs were not shown to activate any of taste receptors. And it's probably at least one reason for this is that the heterologous system is fairly artificial and it may not completely accurately describe properties of taste receptors. So what message we can learn from this complexity? One is that it creates some challenges in using heterologously expressed TAS2Rs for in vitro assessment of drug bitterness, just because there is no universal receptor which would respond to any of the bitter compounds. If you would like to learn more about bitter drugs and which receptors they activate, a very good source of information is called bitter database, BitterDB, and you can find information about the BitterDB on this slide. It was created by Masha Nifin, University of Tel Aviv, and her laboratory. And just an example, this is acetaminophen shown in the table from Meyerhoff paper, and this is data for acetaminophen which have the same citation from Meyerhoff paper, and that it activates human TAS2R39 receptors. And also that there are some other studies which analyzed this bitterness of this drug. Now I will tell a little bit about somatosensory system, which is responsible for perception of touch, temperature, and pain. And one way to visualize how different parts of the body, well, somatosensory system innervates whole our body and represents in the brain information from different parts of the body proportional to how richly this part of the body is innervated. This weird image is called sensory homunculus. And one thing you can notice that oral somatosensory system has rich innervation. It's overrepresented relative to many other types of the body. The other image which to the right, which gives you the same information, showing how much information we can derive from oral cavity relative to other parts of the body. So if you remember our sensory experience, we probably can clearly see that our oral cavity is very sensitive. It is basis for what sometimes is described as mouse feel. And one example to illustrate it, if you imagine like a small object, like say for example hair, and how much you can sense it by keeping it in your fingers, and how much you would sense it if you would find a hair in a meal which you would eat. You will clearly see how much more sensitive the oral cavity is for sensation, for tactile sensations. And the reason for this tactile sensation that both lingual epithelium and hard palate are very richly innervated by somatosensory nerve endings, which have multiple types of somatosensory sensors, broadly distributed among epithelial surface, including taste papilla, like this is image of fungiform taste papilla, which has taste bud and also somatosensory nerve endings. So how does somatosensory system contribute to medicine palatability? Sense of touch is responsible for perception of texture, mouthfeel, grittiness, chalkiness. Temperature can create sensation of burning, warmth, and coolness. And also some chemicals can stimulate the same receptors and generate similar sensations, say contributing to mouthfeel or warmth or coolness. And the next few slides will focus on mechanisms of chemosensation mediated by somatosensory system, their contribution to flavor perception and palatability. The coin term for this type of perceptions, chemesthesis, and it is not as widely known as, say, taste or smell. There are chemosensory field debated how to call this somewhat unusual, less known sense. Alternative terms were proposed: trigeminal chemoreception, chemical irritation, or chemical feel. But in any case, the basis for chemesthesis is trigeminal innervation of the nasal and oral cavities and ability of chemicals to stimulate these nerves, which are also stimulated by pain stimuli. There are a large number of stimuli can activate these nerve endings, including capsaicin, present in pepper, menthol, present in mint, acidity, and some drugs, for example, ibuprofen. And this is a range of sensations generated by chemesthesis, such as cooling, warmth, itch, stinging, burning, tingling. Receptors which mediate this sensation are called transient receptor potential, or TRP channels. They serve dual role, acting as both thermoreceptors and chemoreceptors, and you can see that each of these channels has its own range of temperature sensitivity shown on the x-axis and indicated by color. But also each of them have a chemical which can activate them, and I wanted to give us an example relevant to drugs is ability of ibuprofen to evoke throat irritation through the TRPA1 receptor. This work came from my previous institution, Manel Chemical Senses Center, and my colleagues a while ago have shown that ibuprofen produces sensory irritation of the throat, which often leads to cough. And later, using cellular and genetic approaches and pharmacological approaches, they have shown that these effects of ibuprofen are mediated by TRPA1 channel using both animal and human cells and channels. Now, when we face this question, how to assess medicine palatability, there are a number of ways to do it. Of course, using human sensory tests is the best and most predictable, best approach, which produces the most predictable data, keeping in mind that it's not always or not always easy to test children. But there are many other approaches using in vivo tests, for example, rodent brief access taste aversion or BATE tests, in vitro approaches using human taste cell cultures or heterologous expression of bitter taste receptors, which we discussed in previous slides. In silico approach, for example, docking of receptor ligand interactions, and there are biological and electronic sensors such as amoeba, organism dictyostelium, or electronic tongue. And we don't have time to discuss details of each of this approach. Very briefly, I would mention that each system has its own strengths and limitations, and I would like to highlight the rodent brief access taste aversion test, which is quite versatile and functional system, which we at GSK and some other academic groups and contract research organizations use quite frequently for medicine palatability assessment. One way to look at all the systems for sensory assessment is to ask question, what is exactly measured in these systems? And one aspect of interaction or detection of bitter compounds is interaction with TAS2R receptors. And many of these systems, starting from human sensory tests to in silico docking tests, can use information or materials from TAS2R receptors, can assess this interaction. Some other analog systems, biological or electronic sensors, don't carry task to our receptors, and which may limit their predictability. Now, if you go to the next stage of chemosensory processing, transduction in taste receptor cells, not all systems have all components of taste transduction. For example, cultured taste cells do have this transduction, as well as human and rodents, and more simple systems don't do it. And finally, when we talk about perception, palatability, this is a result of brain processing. So if we really want to measure palatability, the system which has brain is necessary, and it would limit us to in vivo human or animal systems. Well, when can we rely on animal systems, particularly rodents, to predict taste response of humans? The reason for this is that there is good conservation across mammals. If you look at just taste responses, what we can see with our eyes, they are very well conserved. Starting from rats to human babies. They have stereotypical responses of tongue protrusions to sweet stimuli or oral gapes to bitter stimuli. The conservation is also evident if you look at how taste system is organized. Rodents and humans have similar taste receptor cells, similar taste nerves, and brain regions which process taste information. And as an example of this predictable value of rat behavioral test to human perception, this is a set of analysis which our group, including Rob Ives and me, in collaboration with Catherine Toulouse at University College of London, conducted recently and presented last year at EUP5 meeting. Basically, this slide shows on the x-axis thresholds for concentrations at which rats detect show aversion in brief access tests, and the y-axis shows thresholds or concentrations at which humans detect bitterness, and each dot represent each of the 25 bitter compounds. And you can see that the correlation is maybe not perfect, but is pretty strong, and it allows us first to say that rodent tests have pretty reliable predictive value for human perception, and they also give us a tool how to translate through this regression equation thresholds which we obtain in rodents to predicted taste thresholds in humans. This is just some examples of how different groups use rodent brief access tests to evaluate medicine palatability. So when we use brief access tests, it's important to remember to understand what we can and what we cannot measure in this test. And this is a brief access taste aversion test, which is designed to detect aversion. So in other words, it measures hedonic aspects of perception, whether compound or formulation is aversive, neutral, or palatable. It also can assess intensity of this hedonic sensation, but it doesn't directly addresses taste quality perception. For example, if you find aversion, it may or may not be due to bitter taste. It may, for example, be due to irritation, and we can say which modality is responsible for aversion. Now, one important thing to consider when to assess medicine palatability, and this is a very nice compilation of prepared by colleague at GSK, Rob Ives, of what we could learn using medicine palatability assessment at different stages of drug development. I will not go over details of this, just will show another graphical illustration of this process. which is a slide from presentation of at the Smart Pediatric Development Initiative by Catherine Toulouse and Terry Ernst at UCL and GSK. One thing you can notice that brief access test aversion test was recommended to use at several different stages of drug development, and important thing to consider is that there are advantages of starting palatability assessment early, because it first allows more time to respond to any issues with palatability, prepare ways to mitigate it, and also provide some additional possibilities. When drug is early in development, you can choose one or another formulation which may be more suitable in terms of sensory properties. If you detect issue with palatability, how to mitigate it? The three major ways to do it are by concealing or encapsulation using physical approach, and chemical approach using masking and blocking. And the next few slides, which will show a little bit in more detail what is neurophysiological basis for masking and for blocking. So blocking is a pharmacological antagonism to inhibit aversive signal, and it happens when we have beta stimulus or agonist, and we apply antagonist which acts at the same receptor which is activated by agonist. So this interaction happens in the periphery at the level of the taste receptor cell. If we consider masking, for example, one common way to mask aversive taste is by adding sweeteners. You see in this diagram that sweeteners and bitter stimuli activate different taste receptor cells. An interaction between information which comes from these receptor cells actually happens in the brain. So that mechanisms of taste-based masking is central. And similarly, if you want to use olfactory stimuli, for example, pleasant odors for masking, this interaction is also central. So now, how we can map these mechanisms of taste blocking and masking on different systems for sensory assessment. One component which is necessary for assessing blocking is presence of taste receptors, and you can see that some but not all systems are suitable for this assessment, just because not all systems have TAS2Rs, which are receptors responsible for blocking. And if we want to assess masking, which happens centrally, we really need to have test systems which have brain where this interaction occurs. Well, I'm coming to the end of my webinar, and there are a lot of other interesting topics of how taste and medicine interacts, for which I wouldn't have time. I'll just briefly mention them. For example, diseases may affect flavor perception. Drugs may influence taste not by applying when they apply to oral cavity, but also after they circulate in the in our blood or penetrate taste tissues through the blood. They may affect, for example, taste cell turnover. There are side effects of drugs involving taste, and one thing which could be considered when drugs are evaluated, whether their effects on chemical sensation are safety or issues or of target effects. Well, here are a few concluding messages. In terms of chemosensory mechanisms, drug can cause aversion by stimulating bitter taste, chemostatic irritation, and possibly other mechanisms. Our brain combines positive, attractive, and negative aversive sensory information, which provides basis for flavor modification, to reduce drug aversion. Non-human approaches for sensory assessment help to predict palatability issues early in drug development and help to develop mitigation strategies. And one particular test which is quite versatile in addressing these issues is rodent brief access taste aversion test. To mitigate palatability issues, blocking and masking approaches could be taken, but if you want to evaluate them, different sensory assessments would need to be used. Important to remember that brief access tests could be useful for evaluating both blocking and masking. And finally, these are just a few things which you may recognize from previous slides. Should remember that flavor and odor is not the same. Bitter is not always aversive, and vice versa. Aversive is not always bitter. Children differ from adults in how they perceive and respond to aversive stimuli. And it is advantageous to start medicine palatability assessment early. It is better to detect problems in preclinical rather than in clinical setting. And finally, I would like to acknowledge several people who helped me to craft this presentation, which helped me to figure out what exactly questions which would be interesting to the drug formulation audience and what answers you are looking for. And with this, I'd like to thank you for your attention, and I'm ready to answer questions. Thank you.
Thank you very much, Alex, for the wonderful presentation. So I'll start with one question from Trupti Dixit. Wonderful presentation. And how does Bata correlates with pediatric taste?
Well, we unfortunately we don't really have data which would answer this question. We can guess, but really to answer this question, a study would need to be designed, and I hope that my inability to answer this question may help to drive some interest to actually conduct studies like this. I hope that in June at the IQConsortium workshop, we will be able to discuss and maybe develop plans to address this question.
Okay. Thank you. So next question I have from Alex Wu. I'll start with a comment from Alex first. I think greatest bitterness neuroscience talk I ever heard recently. And the question is, what is exact binding location for blockers in TES2R receptors? Can you go back to that page?
I'll try. I think we need to ask Mahali because she has control over screen now. Probably I'll try to answer in my just orally that the binding sites for, if I understand the question correctly, binding sites for different ligands of bitter taste receptors and for different blockers of taste receptors, I expect it to be not the same. The bitter taste receptors, bitter taste receptor agonists were shown to bind both, right, it's correct slide, both extracellular and transmembrane and extracellular domains, and different bitter blockers would likely bind unique places. There were very few studies which did site-directed mutagenesis to address this question, and maybe offline I could dig this information and share it. By the way, feel free to follow up with any questions, or by reaching me directly, you should be able to find my contact information.
Thank you. Next question from Daniel Reed. Are there any human taste cell lines that could be used to find bitter antagonists?
Well, this is a question to which Danny Reed probably has better answers than I do. Danny Reed is my colleague at Manel, and I know that she is leading a team of Manel scientists which collaborates with a biotech company which actually develop human cell lines responsive to bitter taste, which could be used for these purposes. The other company which reported screening systems using human taste cell lines is a German company named Brain.
Okay.
The company is Discovery Biomed. Sorry, I had a senior moment. Manel Center and Discovery Biomed, actually funded by NIH to develop bitter responsive cell lines.
Okay, thank you. I have a next question from Dennis Murphy from Merck. Is there a correlation between receptor type and expression with taste preference between children and adults? Example, are different test 2Rs expressed at different levels in children and adults?
Frankly, I don't think anybody has conducted this kind of analysis. My kind of semi-intuitive feeling, which may or may not be wrong, is that probably the way bitter taste receptor cells are composed pretty mature in newborn children because it's important for them to detect bitter. And what we know from different studies that each bitter taste receptor cells express multiple or maybe all or almost all bitter taste receptors. So this feature of single cell expressing multiple bitter taste receptors most likely is very similar in children and in humans. And where difference occurs, I think mostly it occurs through exposures and through learning, because while children are typically uniformly respond to bitter taste with aversion, humans learn that not all bitter compounds are harmful, and they learn to consume these sources of food or beverage and tolerate bitterness. So whether level of expression or patterns of expression of bitter taste receptors differ in humans and children, my personal bet would be that probably not, but I don't have solid data to substantiate this claim, and I think that there may be some scientists who may disagree with me. So I would say this is a question open to debate.
Okay, thank you. And one more question from David Clapham. Is there any correlation between the persistence of binding to the T2R receptor and the perceived persistence of the taste?
Right. So temporal aspects of taste. Yeah, I didn't have chance to discuss them, but they are very important for both taste causing aversion and taste causing attraction, using to mitigate bitterness like sweeteners. And I guess when we the kind of common phenomenon or problem is lingering bitter taste, and I think there may be several factors which could contribute to this lingering bitter taste. One is binding to receptor, and I guess theoretically probably the constants of dissociation for different receptor ligands would vary. I just don't have specific information or specific data which would specifically assess this kind of rate of dissociation. So basically binding to receptors, or how strongly ligand binds to receptor, probably would contribute to temporal aspects of taste. But we also should remember that bitter compounds could bind not only to receptors but to other tissues. They, for example, can get stuck into cell membrane or can get attached to extracellular proteins or to like proline-rich proteins, something which is not taste receptor itself, but something which would keep bitter taste compounds or stimuli in the vicinity of receptor, which will create some locally elevated concentration. Some bitter compounds are lipophilic and they can get inside of the cells, and this may contribute to their lingering taste. And finally, as I mentioned, some drugs can quickly get in circulation and reach taste receptors not only through the saliva, through the oral cavity, but also through the blood and continue causing taste sensations. So it's probably quite complex, important, serious problem, and probably a lot of different contributors to lingering aspects of bitter taste.
Okay, well, one more question from Kyle Palmer. Would you expect blocking strategies to result in different outcomes between adults and children?
Well, I guess based on the theory which I exercised before, that first that blocking occurs at the level of taste receptor cells, and second that my guess is that taste receptor cells in children and adults are similarly mature and express similar repertoires of bitter taste receptors. I would, my hypothesis would be that blockers should be equally potent in children and adults. I don't have any data to support this hypothesis, so.
Okay, with that, it comes to the end of the presentation. Thank you very much to Dr. Alex Bashmanov for the wonderful presentation. And with that, I'll pass back to Mahalia to wrap up the webinar.
Thanks.
Thank you again.
Thank you, Alex. That was really an incredible presentation. Thank you so much for the time that you took to prepare it. Once again, I'd like the audience to know that this webinar has been recorded, and we will be circulating a copy of the recording to those who attended today, and an invitation to attend an on-demand version of this webinar for your colleagues. We'll also circulate the slides for your reference. And one last reminder to please complete the feedback survey. We'd like to continue to provide these types of informative and valuable webinars to our IQ member audience as well as to the public. So it'll take just a couple of minutes, and we'd really appreciate your feedback. And until then, we do have a June webinar scheduled. We hope that you'll join us, and we look forward to seeing you all online then. Thanks very much, everyone. Thanks again to David, Beth, and to Alex. Have a great day.